Think about your cells. You've got lots of cells, right? You've got brain cells and skin cells. You also have cells in your gut. Not just your own cells, the cells that make up your intestines and your stomach, but also bacteria cells living in your body. Well, have you ever thought, why are these bacteria so small? Why are they microscopic? In fact, we call those bacteria the microbiome. Why are they so small? Why have they stayed so small through evolution? Could they get bigger? Could bacteria like E. coli ever be as big as, say, a whale? That would be pretty scary. A big single-celled blob walking around or living in an ocean. Could that happen? Could single cells ever become as large as a whale? In this podcast, we're going to tackle that question. How big can a cell get? Let's get started. So we want to start by talking about a few characteristics that all cells have. Whether it be a bacterial cell or an animal cell, all cells have these four characteristics. So all cells are surrounded by a plasma membrane. This is a membrane around, here you can see, and there you can see it in the animal cell, that protects and keeps things inside. It is semi-permeable, meaning some things can permeate it, some things can't. All cells also have some sort of cytosol. Think of that semi-fluid substance inside the cell that you see there. Now, you may have also heard the word cytoplasm. So these words are used a lot interchangeably, but technically, cytoplasm would be the cytosol, that semi-fluid, plus the organelles that are inside it. So all cells have a membrane. They have cytosol. All cells also have some genetic information. Here in the bacterial cell, you'll see that the DNA is just kind of hanging out there in the center. Over here in the animal cell, you'll notice that the DNA is wrapped very well and protected in the nucleus. Okay, so the fourth thing that all cells have would be ribosomes. Well, what do ribosomes do? They make proteins. They synthesize proteins. So since all cells need proteins, all cells will need ribosomes. So there you see ribosomes in a bacterial cell and ribosomes here in a nice animal cell. So all cells have all four of these characteristics, whether you're a bacteria or an animal cell. So you've seen me talking about these two cells, bacteria and animal cells. We actually want to be a little more specific. These fall into two classes or categories of cells. Bacteria actually are prokaryotic cells. So you think of pro like before, like a prototype, right, early on. Prokaryotic cells, these evolved first. These are sort of ancient cells that evolved a long time ago. And they have DNA in a nucleoid region. So here, it's not a true nucleus, right? There's no membrane around it. It's just a region where the DNA or the nucleic acids hang out. No other membrane-bound organelles. They do have ribosomes, but those aren't bound by a membrane. Prokaryotic cells also have a cell wall. So in addition to the plasma membrane that you see there, they have these cell walls that surround the membrane. Two examples, the two domains archaea and bacteria, fall into the prokaryotic type of cell. Okay, so prokaryotic, think early on, then eukaryotic, or a true cell that has all these organelles. So animal cells, plant cells, fungi, right? These all fall into the eukaryotic cell classification. And they, of course, will have their DNA in the nucleus, surrounded and protected there in the nucleus. And they have other membrane-bound organelles. The mitochondria, probably your favorite. Lots of other organelles, endoplasmic reticulum. And these, what we'll find is they're much larger than prokaryotes. So these bacteria are much smaller than the animal cells, plant cells, fungal cells. Just to show you them, they look about the same size, but these over here are much larger than the bacterial cells. So back to our original question, what limits cell size? It is actually all about the surface area and volume. Those two compared make up the surface area to volume ratio. And what this means is that as a cell gets bigger, the volume increases faster than the surface area. So thus we need small cells because there's a greater surface area to volume ratio. Now that's a lot of words. Let me give you an example. Take here, let's just pretend we have a simple cuboidal cell, a simple cell that's a cube. Okay, we're going to use this because it's easy to do the math. So let's say it's 1 centimeter. Now this is obviously, cells are not that big. So we're just using this for an example. So let's say it's a 1 centimeter cell. So you got a 1 centimeter there, right, by 1, everything's 1. So if you think about the surface area, think of area, length times width, right? So 1 times 1, this would be a surface area of 1. This would be a surface area of 1. 1 on the bottom is 1. On this side is 1. On the back. All those together would be 6. A surface area of 6 centimeters squared. Now if you take the volume, length times width times height, 1 times 1 times 1 is still 1. So that gives us a surface area to volume ratio of 6 to 1. So that small cell, 1 centimeter, let's say, has a 6 to 1 ratio. Now, let's say a cell gets bigger, and we're just going to use some simple math here again. Let's say it's a 2 centimeter cell, 1 by 1, 1, 2 total. So we've got 2 by 2 to get surface area, length times width. So we got a 4 there, we got a 4 there, we got a 4 there, 4, 4, 4. All those added together, 24 centimeters squared. The volume, length times width times height, would be 8. So if we look at that ratio, now we're down to a 3 to 1 ratio. So with smaller cells, the ratio is bigger for the surface area to volume. Surface area to volume here is just 3 to 1. The surface area has been cut in half, essentially. The surface area to volume ratio has been cut in half with this group. Let's do one more just to show you. Let's say that we got a 3 by 3 now. Okay? So a 3 by 3 surface area would be 54 because 3 times 3 is 9. Length times width for this whole surface area. We got a 9 surface area there. We got a 9 there. And then on the back and the back and the back, all those 54 centimeters squared for the surface area. Now if you do the volume, length times width times height, you've got 27. So now notice, when we get bigger, the surface area to volume goes down even more. Here we're looking at a 6 to 1 ratio. Here it's just 2 to 1. So who cares? Why does the surface area to volume ratio matter? Well, it's actually all about metabolism. So if you think about metabolism, right? Breaking down food, bringing in food, building up parts of the body in the cell, all of those requirements need surface area because everything has to move across that semi-permeable membrane. So when we have a lot of membrane, a lot of cell surface, things can go in and out. So let's think, what needs to go in and out? Well, obviously we need some oxygen in the cells, right? A byproduct might be CO2, right? Breathe in O2, at least we do as animals. Breathe out CO2, that needs to go out. We need to bring in some protein, some amino acids. See that nice amino acid there, and the amino and the acid, that needs to go in to make our proteins. We need sugar. We need some carbs to come into the cell. So lots coming in, lots going out. CO2, here's some ammonia coming out. Now, in this single cell, right, there's not a lot that can fit inside because of the volume is relatively close to the surface area. But look at over here. This is a bigger cell, and you may say, well, sure, there's a lot of surface area there. Things can go in and out. But there's so much more volume in the middle now with this larger cell that it is inefficient for all these things to go in and out. So the surface area must be large enough to exchange materials. Otherwise, as much stuff can't get in and out as needs to, because cells need to get their nutrients and eliminate waste, and if there's not a lot of surface area, they can't. Think of this sort of like a large stadium versus a smaller room. It would be much harder, a lot more people can fit into a large stadium. It would be a lot harder to evacuate that stadium as it would be to evacuate a smaller room, just because this one's so voluminous, it can hold so many things or people. This, it's fewer things it can hold. So then, how did plants, trees get bigger, right? Because you see big things around. Think of a whale, think of a giraffe, think of a large tree. They're obviously bigger than small single organisms. How did they do that? Well, what we evolved to do is become multicellular. So instead of one big cell, you have multiple, 1, 2, 3, 4, 5, multi-cells, each with their own plasma membrane. So think of how much more membrane is here than here, right? If you were to trace this around and measure the circumference, and then trace all these individual ones and measure their circumference, you get a lot more surface area when we divide and become multicellular. So, will there ever be a huge bacteria, single-celled bacteria roaming around? No, because it's just not possible for it to maintain metabolism with all of the specific things it needs to do in the surface area. So, if you were ever asked to calculate the surface area to volume ratio, all you got to do is look up what the surface area and volume is. Because we have lots of different types of cells. We got cells that look like spheres or cubes, rectangles, cylinders, right? Lots of different shapes of cells. So you might be asked to calculate the surface area to volume ratio for any of these cells. And it would be simple, right? All you'd have to do is calculate the surface area, calculate the volume, and then if you have to compare it to another one, just compare it. So let's just do a simple one. Let's say we've got a sphere with a radius of 2, and we want to calculate a cube with a side of 2, okay? So let's say here that 4 pi r squared, so we got radius, we'd put a 4 there, so 4 pi 4. The volume would be 4/3 pi r cubed, which would be 2 times 2 times 2. So that would be 8. Okay? So you could calculate what those are and get rid of the pi's, right? And then calculate over here with the sides, 6 times 4, 2 cubed, and then compare those to each other if that was the case. So no need to memorize these. You can always look these up. These will be in some handout, or you can always look these up online if you need to. So, what are we going to do for the rest of this podcast? We're going to start going over these individual organelles. Well, the same reason, or for one of the same reasons, that cells can be only so big, right, because of the surface area to volume ratio, higher classes of cells, like animal cells and plant cells, started to divide up the work so that they can have internal membranes as well. So a nucleus has its own membrane. A mitochondria has its own membrane. Endoplasmic reticulum has its own membrane. More membranes mean more things can move across. So eukaryotic cells divide up their work, right? It's got special organelles that have specific functions that divides up the work, and it's a lot more efficient. So what we're going to do in the rest of this video and then the following video is I'm going to go through these organelles, and we're going to talk about what each one does and what it looks like, what its structure is, and what its function is. So we're going to start that in this video, and then we'll continue it in the next video. So let's start with the nucleus. One of the most famous of all the organelles, right? So let's zoom in, and what we'll find about the nucleus is that its structure is a double membrane. It has this nuclear envelope, right? It envelops or is an envelope around the nucleus. And this double membrane is fused with these spots that are called pores, nuclear pores that allow things to move in and out. So you also have a very center part of the nucleus, maybe you could call it the nucleus of the nucleus, that is called the nucleolus. And this is where ribosomal RNA is made. That's the RNA, of course, that makes up our ribosomes. So that's the structure, double membrane. It's got some pores so things can go in and out. What does it do? Right? Well, its function is to contain and protect the DNA. So this is where your DNA, right, that genetic material, those nucleic acids are found. It is also the site of transcription. So we'll see that this DNA later will become messenger RNA, which will then leave the nucleus. And I also mentioned, of course, ribosomal RNA is made there in the nucleolus. Where does the word nucleic acid, where does the word nucleus come from? Oh, I gave you the answer, right? It contains the nucleic acids, DNA, and RNA. So that's the nucleus. Ribosomes. I mentioned this one before as well. Ribosomes, you can see them right there. There are ribosomes that are the site of protein synthesis. Now, if you take a look, we've got some ribosomes that are bound, all right? And then we've got some that are free floating. So if we take a ribosome and we really look at it, what we'll find is that the structure, it contains ribosomal RNA and some protein. Okay, so that makes sense. Ribosomes are made of ribosomal RNA and some protein. And there are two subunits. You've got the large subunit up here and the, guess what, small subunit down there. So what do? I've mentioned this before. Their function is protein production. Ribosomes are the site of protein production. So what you'll see is that there are different ribosomes, and I'll talk about that. And what we find is that these two types of ribosomes, we see the bound ribosomes that are attached to the endoplasmic reticulum, and these make proteins that probably will need to go outside of the cell or they're bound for the membrane. Then you also have, as you see there, some free ribosomes that are in the cytosol. These will make proteins that we generally will keep within the cytosol. Okay? So all ribosomes make protein, but the ribosomes on the ER, the endoplasmic reticulum, will probably be exported or go to the membrane. The ones just hanging out in the cytosol, the proteins that they make will also function within the cytosol and probably won't be exported. So, as we wrap things up, we want to talk about these last two that I'll get to in this video. That's the rough endoplasmic reticulum and the smooth. So I've mentioned the endoplasmic reticulum, right? They're all around there. What we see is that some of it is rough, meaning it has these ribosomes on it, and some of it is smooth. No ribosomes attached. So let's start over here with the rough. The rough ER structure is basically just some membranes connected to the nuclear envelope, and it has ribosomes embedded. Okay, there's all the ribosomes. That's why it's rough. It's got the ribosomes. What does it do? Well, it helps compartmentalize the cell, right? Because it has so many membranes, it can divide up the cell into these little compartments. And it's the site for protein synthesis because it has the ribosomes on it. We also will find that the rough ER also makes or manufactures some membranes. So rough ER, think all about compartmentalization, and it has the ribosome, so it makes the protein. So what's going on over here with the smooth ER? Notice that there are no ribosomes on that one, right? No ribosomes. It's smooth. So its structure is similar to the Refiar except without ribosomes. It's just these flattened membrane sacs. Its function is to make lipids and also detoxification. So what we find is that there's a lot of smooth ER in the liver because the liver is involved in detoxification. Okay, so we've got rough ER, we've got smooth ER, endoplasmic reticulum. The rough has the ribosomes, synthesizes proteins. The smooth ER, no ribosomes, and is involved in detoxification and lipid synthesis. So this was just an introduction to parts of the cell. In the next video, a tour of the cell part 2, we'll go over the rest of the organelles in the eukaryotic cells.